For today's episode, we will cover a fascinating and very powerful phenomena, visual thinking. We will cover the pictures and the movies playing in our head. We will cover lots of brain regions and connections, starting with eye biology. This topic won't be exhaustive, but we will get into some deep and fun areas. Remember, eyes are brain and bidirectional influence on our central nervous system, or more specifically, the autonomic nervous system. We will cover how sensations turn into perceptions, and also think sensory processing. There are 2 previous episodes solely on sensory processing. The sensory processing phenomena, all autistics have a sensory processing challenge. That rate of information coming in. And think about eye contact. Now, it shouldn't be that difficult to understand why eye contact is not preferred. And think about the references to Bee Movie, the animated movie about bees. Early in the movie, when the teenage bees are graduating high school and they're kind of thinking about life, and the cameras zoom out, looking at the hive, and there are thousands upon thousands and thousands of bees swarming around at high rates, very efficient, close together. Everything is in sync. They are extremely efficient, and nothing is abnormal in this whole process. That's visual thinking to me. I want to start with an anecdotal event that recently just happened about visual thinking. For the context, I guess, this is in a graduate school classroom. We were talking about visual perception and visual thinking, visual hallucinations and so forth. And I was asked about this. And the question was asked, well, how do I know I have this? The person was asking how can they tell if they have this? And that's very obvious. Like, you'll know. So the professor said, if I say tree, do you see it? Now, here's the thing. I will explain my experience with it, and this happened fast. I instantly went to tree bark, oak tree specifically, because of the rigidness. There's distinct lines in tree bark, and I saw this in real time from about 3 or 4 feet away. I could see the distinct features of the oak tree, where there's almost vertical line with the rough, kind of rigid bark sticking out, and then there's the recessed lines, there's the vertical lines that are often smooth. And I walked right up to it, and I put my left hand to rub across the tree bark, and then the right hand with the index finger to rub up and down the vertical lines. Now, I could feel this, and I could feel this. I could feel the touch signals as if I was actually touching the oak tree. The left hand with the ruggedness, and then the right hand up and down the lines. And I could feel it through my heart and body, as if I was really touching the tree. And then my mind went into this rapid categorization of different tree barks. An oak tree that is less distinguished with the vertical lines. Sometimes they have vertical lines, sometimes they don't. And there's different types of oak trees that showcase this. And then from here, the pictures just came rapidly through my head. Different types of kind of rough tree bark, all the way to the smooth type. Now, there's things like Bradford pear trees that are rough, but there are other types of pear trees that are smooth. And then eventually I got to a magnolia tree, which is my favorite type of tree. Now, in the matter of a few seconds, maybe 8-10 seconds, I saw 10, 12 types of trees. The bark, this is the thing, I'd never even made it to the canopy. I just went into this rapid categorization of tree bark and the tree trunks. Now, visual thinking and seeing these movies play out is one of the most fascinating things of my life, and I think with the autistic phenotype. So we're going to cover this in 2 parts. The next episode will be about machine learning that's similar to AI, and how we build catalogs for different categories. This is the source of accelerated learning and those kind of restricted fixated interest, and how it's just like a filing cabinet with vast amount of information, like AI. Okay, so now let's get back to the intent and material of this episode. I'm finished with the anecdotal. Now let's get to the material, the information regarding visual thinking. If you think about if you have a destination and there's point A to point B, but there are 5 stops that you must get to. For autistics, these first 2 stops are going to come fast, and the terrain is going to be rocky. It's going to be tough. For non-autistics, point A to point B might seem just like a normal venture. Nothing outstanding or remarkable to get from point A to point B and to execute these 5 stops. Now, by the time you get to the 5th stop with the autistic, things are complicated on the other end. Let's say for the autistic phenotype and the sensory processing, the first 2 stops come fast, but then stops 3, 4, 5 to the final destination is lagging versus the typicals. This is just analogy with eye biology to reaching the cortex. We have all of these stops in these brain regions, all of these relay stations and hubs inside the brain that are working together, orchestrating and facilitating adaptive responses, so to speak. The central nervous system is designed to move the living organism, and the sensations coming in, extracting physical phenomena from the outside world, are used to inform the living organism of the environment. Think about the mesencephalon, the superior colliculus as well, and how this is just complicating everything with this relay, recruiting those distal connections throughout the brain, and especially the cortex and the prefrontal cortex. Those are distal connections. So the eye biology, how do we send sensory neurons into interneurons? Remember the previous episodes with the parvalbuum interneuron, and then into the motor neurons. So we have those 3 types of basic neurons: sensory, interneuron, and motor neurons. So that we can form perceptions of the world. Where do we bias our attention and orient to the external world? Or the fantastic interworld, often described in the autistic phenotype. Remember, autistic, autism means self. We will also highlight the social back and forth, troubles, and keeping up in real time, a source of echolalia as well. And we will also mention the reading process. And we will cover lots of Temple Grandin, 3 specific books. We will just extract information from these 3 books. I'm always fascinated with thinking in pictures. For Grandin writes, I think in pictures. Words are like a second language to me. I translate both spoken and written words into full-color movies, complete with sound, which run like VCR tape in my head. Temple Grandin is a pioneer for this type of research. And remember, a key takeaway here, this is crucial. Temple Grandin is from the inside. Get it? Autism is mostly interpreted from the outside, from non-autistics. Huge problem. We can sit here for days, months, and years and say things like mitochondrial dysfunction or the cytokines, gastrointestinal problems, those poor distal connections described earlier with point A to point B, but bringing the autistic phenotype into life is needed too. And finally, we will once again mention oxytocin and cover some Kanner and Asperger kids. So let's start with early eye biology and expand on the magnocellular and parvocellular cells and pathways. Again, from Temple Grandin, she says, sensory processing problems are real. Bright lights can be overwhelming, like a spotlight in my eye. So this points to eye biology, where visual thinking begins. So with the retina, this is a light-sensitive layer and acts as a camera lens, converting light into signals via retinal ganglion cells. There are different types of retinal ganglion cells, the rods and cones, and then the popular melanopsin. These form the optic nerve that connects to the lateral geniculate nucleus in the thalamus. This is deep inside the brain, right next to the mesencephalon, the midbrain, so to speak, which isn't exactly the midbrain. Maybe 50,000 years ago, it was the midbrain. This is wired up in the womb to shape how we see. In autism, there are denser retinal ganglion cell connections, possibly from a mutation that we've discussed called SHANK3. And this amplifies inputs, making those examples that Temple Grandin shares like razor sharp, but sometimes overwhelming. The melanopsin containing retinal ganglion cells, intrinsic photosensitive retinal ganglion cells, they are sensitive to blue light, and this regulates alertness. And the dysregulation may heighten the light sensitivity. Melanin, a retinal pigment, melanin is kind of much more than a pigment, facilitates signal transfer. Remember the first episode of the sensory processing episode where we talk about melanin. Melanin sits behind all of the sensory processing organs, all of these receptors from the outside world used for sensation. Melanin is behind this. And this connectivity, the autistic phenotype has hyperconnectivity of the optic nerve projections to the lateral geniculate and the superior colliculus. Remember, these have spatial relationships. These are very close. They can reach out and basically high-five each other. But the superior colliculus is like a relay station. It acts to recruit distal connections. And there's an integration system here with the superior temporal sulcus, STS. This is the first time we've mentioned this on the podcast. We will get into a little bit more of that momentarily. But from the retina, these familiar Magnocellular, sometimes M pathway, and Parvocellular, sometimes called P pathways, are like editors. The Magnocellular neurons are big and fast, and they spot motion and spatial patterns. The Parvocellular neurons are small. They focus on details and color. The Magnocellular is colorblind. However, the Parvocellulars are picking out fine detail and colors. In autism, the M pathways are hypersensitive to boosting dynamic imagery, whereas the Parvocellular acts to highlight the fine details in objects, visual inputs. The Magnocellular neurons use more of a fast glutamate signaling, excitation, whereas the Parvocellular neurons rely more on GABA, inhibition, so more of this EI stuff. The M pathways project from the lateral geniculate nucleus to V1 and to the parietal lobe, a higher order sensory integration area of the brain. But specifically here, we're going to talk more about the precuneus, which is huge in the visual thinking phenomena. And in autism, there are hyperconnected short-range fibers that enhance the spatial imagery. The P pathways project to V1 and the STS. But, and here's a big thing here, it's underconnective, hypoconnective via inferior longitudinal fasciculus. Now, if you remember the speech and language episode, we talked about the arcuate fasciculus in quite detail. Fasciculus, there are bundles of nerve fibers, axons, within the different brain white matter areas. And this is connecting different areas of gray matter, acting as communication pathways. This is very huge here with these distal connection problems. Now, there are different type of fasciculi, just depending on their roles, what they're trying to accomplish, such as the arcuate is for speech and language, so Wernicke's and Broca. The inferior longitudinal fasciculus is a major thing here for the white matter tract in the brain's visual pathway, which is connecting the occipital and temporal lobes. And this is helping process and integrate visual information with those higher level cognitive functions, including object recognition or the face processing. Now, I just mentioned the temporal lobe and the face processing. There's a cool nuclei called fusiform gyrus, which is pretty researched in autism. This is facial recognition. In addition, for the language comprehension. Now, if you think about reading, okay, you're using your visual inputs to make sense of the words on the paper or whatever you're reading from. And then it even makes sense for the arcuate fasciculus to come on board because you're reading, making sense of language. And while reading, you're silently speaking to the self. Anyways, different topic. So far, the main things to remember are there's disturbances in these distal connections. Remember the types of fasciculi. And then the point A to point B with those 5 stops is basically inputting sensations from the environment is point A, and then you're trying to get them back to the cortex to make sense of the things that you're extracting from the environment. That's point B, the final destination. So you can incorporate these things and make decisions to move the living organism. How are you going to bias your attention to the outside world? So now I think we can talk more about some neurobiology of these visual inputs, these the visual regions of the brain. So earlier I mentioned visual cortex in V1. And this is at the back of the brain. This is the camera capturing the raw visual data that are being sent through from the retina to the thalamus and superior colliculi. The primary visual cortex, V1, decodes edges and contrast, like spotting a line on a blueprint. Now, I'll have an anecdotal story about blueprints, maybe in the next episode on visual thinking with the machine learning and AI. However, for now, there are also secondary areas, V2, V3, and V4, and this is building objects. In autism, data show that V1 through V4 are hyperactive, and these firing neurons are like a super HD camera zooming in on tiny details. Remember, we've talked about this, how we see details and then general. We build the model from the specific details, fine details. And these can be studied with EEG and the N100 type of measurement machine, MEG and the M100 measuring machine, or fMRI is probably more familiar. And this can reveal increased firing rates that boost contrasting sensitivity. This is why I believe that Temple Grandin in the autistic brain says that she notices flaws others miss. She's very good at this with her profession in the animal industry. This reduced synaptic pruning leaves dense dendritic connections. Remember, we've talked about pruning, like serotonin in the thalamus is pruning those dendrites in the thalamus and the cortex that map out sensory processing. The serotonin is building our sensory map. We've talked about things like that in the SHANK3 and P10. So if you think about a garden or farming fields, and they're neat rows across the field. Just imagine if there weren't neat rows and everything was structured and organized. And it's just Mother Nature just kind of took over and there was no trimming. and it was kind of hard to see the plants in that situation. A region of interest here is from the previous episode with the Parvocellular interneuron inhibition. These are acting as like traffic controllers. They help organize stop and go. Remember, excitation is activating, so that's the go, and the inhibition is inactivating. So that's the stop, the no-go. And these Parvocellular interneurons, because their job is to stop, because there's a lack of stop, there's an amplification of excitability, sometimes causing sensory overload. Remember the pioneer study from Rubinstein, not sure, and Mike Merzenich from 2003. It's a very well-known study in autism on the EI imbalance. So the hyperconnectivity within the V1 through V4 and these short-range occipital fibers, this strengthens the detail processing, like a fast Wi-Fi network, if you will. But the weak long-range connections to the prefrontal cortex via a subtype of fasciculus called the inferior fronto-occipital fasciculus, IFOF. This limits abstract integration. Okay, this is a huge thing with the autistic phenotype. Abstract thinking. Also involved here is that parietal lobe, specifically the precuneus. Now, the second guest on the episode, Dr. Leanna Hernandez from UCLA, briefly mentioned the precuneus having abnormal connectivity with the precuneus. And we've later talked about that more specifically with the default mode network and with the visual thinking. This is in the upper back part of the cortex, the back third of your head, top of your head. The precuneus is essentially like a director in this visual thinking and these movie analogies. And it sequences the frames, if you will, into dynamic scenes. Now, if you've ever heard Temple Grandin be able to describe how she can just see the outline and the layout and the operation of these kind of animal machines, these cow pathways that she's designed. She's famous for these designs with the animals, specifically the cows. She can see this and build this and highlight all of the malfunctions and potentials of error, the bottlenecks and the problems that the cows might face while navigating that point A, entering these chutes to the final destination, the point B. She can see this and kind of modify these designs in her head with the visual thinking. And here, using the fMRI as well, this shows hyperactivity with the autistic phenotype during visual spatial task. And this really enables vivid simulations that take place in this visual thinking realm. However, the fMRI shows a hypoactivity in social task, like imagining someone's perspective. So reduced GABAergic inhibition, so remember the Parvocellular interneurons, combined with an increased so-called mini-column density. These are tiny neural processors, and this amplifies imagery. So we have not discussed many columns so far. Not once, I don't think, in the whole podcast. So we're going to do that now. Many columns are vertical arrays of neurons in the cerebral cortex, and these are organized as functional units that processes information in a highly coordinated way. These are specialized columns, specialized regions, with primary roles of integrating sensory and providing cognitive and motor information. Each mini-column contains approximately 80 to 100 neurons, including excitatory pyramidal neurons and inhibitory interneurons, neurons, such as the parvocellular. These are arranged in a narrow column that spans the cortex, 6 layers. Remember, the cortex has 6 layers, and they're essentially stacked up like crepes. The many columns are critical for processing sensory input, such as this visual thinking, and the pattern recognition, and the detail-oriented cognition that we mentioned. They serve as a basic computational unit for the cortex, and each of these many columns handles specific features. The many columns are particularly relevant to autism due to their role in sensory processing and the pattern recognition. Data have shown that the structural and functional differences in many columns in autistic brains impact visual thinking and the related task. So with the increased density, this leads to denser neural packing, enhancing local processing, and then the reduced inhibition, those fewer stop signals between the many columns for the autistic phenotype can result into less filtering of sensory input. So there's an overload here. And this leads to that heightened. Remember, we use the word heightened in the sensory processing. This details the perception. With the autistic phenotype, these many columns in the visual cortex, V1 through V4, and the parietal lobe show hyperconnectivity with local regions. Remember, the point A to point B is fast, so-called and fast and tough, rigid. This supports that vivid imagery. Some mechanisms with the neurobiology and these many columns, because these are highlighted in several brain regions. The visual cortex, the V1 through V4. Many columns here process those visual features, and this contributes to the responses shown in the N100 and M100 data. And this supports the autistic's enhanced visual thinking. In the precuneus, the many columns here handle visual spatial imagery. And this enables mental rotation and dynamic imagery to formulate and just light up this area. The parvocellular interneurons, well, these regulate many column activity. But the reduced inhibition in autism enhances that local processing. So if there's no stop signals in these areas, there's going to be an amplification of the visual detail and the focus. And the default mode network, and these interact with integrating visual imagery with memory. And this is help capturing the detailed memory and the preference for just having this imaginative world inside of us. However, it can also be a source of things like just rumination and just reflecting back on previous moments, especially those social processing experiences. We can just replay this and just kind of destroy ourselves because of this analytic tendency that the autistic phenotype will have. Okay, so we were discussing different brain regions, and we were highlighting the precuneus here. Let's move on to the prefrontal cortex. This is at the front. And this is kind of like a flexible rule-setting machine that we have. And in autism, with the visual thinking, the dorsolateral prefrontal cortex is a region of interest. And this is trying to visualize goals. In autism, there's a reduced top-down control, and this kind of so-called abnormal abstract thinking. These are categorized by the images, not abstract ideas. fMRI shows that the dorsolateral hypoactivation in abstract task, and there's an atypical myelination in the superior longitudinal fasciculus that disrupts this efficiency. This is like a delivery station. You want these to hit on fast and efficient temporal components here. So of course it's not surprising that this is limiting the top-down control. and the adaptive responses. The superior temporal sulcus, mentioned earlier. This is along the brain sides, and the STS is the sound mixer, if you will, for these visual thinking and movies. And this is blending visuals with sounds and touch. In autism, there's a reduced activation that limits this, making imagery sometimes silent. Desynchronized multisensory neuron firing. That's due to the low parvalbumin function, and this disrupts integration. The reduced arcuate fasciculus and inferior longitudinal fasciculus connectivity impairs multisensory processing, like reading or reading out loud. Transposing this raw data coming in, either visually or reading, whatever sensation it is. This is kind of a delayed process because we have to shift everything. We have to create everything into these images. And sometimes they can even come alive. These movies that we see, real life movies going on in the inside. Now, in the autistic brain, another one of Grandin's books, she writes, the autistic brain is different, but it's not broken. It's just wired for strengths, like visual thinking. Previously, we spent a lot of time on how kind of society doesn't really mesh with the autistic phenotype here. We're very much in contrast with the generalization of social norms and central education and employment and so forth. However, I really want to highlight the Canner kids and Asperger kids. These are always salient and crucial topics to discuss for autism. In 1943, Canner described children with social isolation, the language delays, repetitive behaviors, and visual memory strengths, like memorizing patterns. If you remember what we talked about with the hyperactivity in the visual cortex and precuneus, with the enhanced Magnocellular Pathways, and the enabling of the vivid imagery, this is very much in line here. Now, there's no way to understand the brain functions or imaging the brains of the Canner kids or the Asperger kids, but the behaviors that they highlighted very much is in line with visual thinking processes. The Canner kids, it was really fascinating that it would always appear that they weren't paying attention, but if you'd ask them about the environment, they could pick up and tell you essentially everything happening. That fast visual processing just allows us to detect things in the environment much faster than others. Canner kids are like photographers. They're just capturing every moment, every detail. But the trade-off is there's no interaction. Maybe because people are just not on the same page. If you don't experience the same thing, how are you going to share that? How are you going to share those common interests? If you think about criteria A and the DSM, social interaction and communication criteria, there's that implications with the back and forth. But if you get autistics on a topic, or autistics comfortable sharing something, there's massive amounts of interaction. In 1944, Asperger described that the so-called higher functioning children with preserved language had intense visual technical interest. One common example from Asperger's case studies is Fritz. Asperger's little professors, and he highlighted that so-called autistic intelligence. They're technical wizards here. They're so technically advanced and just picking up patterns and picking up small details in the environment and having these fixated interest on these types of things. Canner and Asperger's observations kind of really capture why the word autism or autistic is used. That inward focus, mainly because the amount of intensity and enjoyment, the interest with this visual thinking, and that safe place of these topics and subjects. Something shared across the so-called autistic phenotype. This is the core, that B3, with the restricted fixated interest, that are abnormal in intensity or focus. Now, something we haven't discussed yet, and it's very powerful, is the default mode network here, how this is playing and kind of helping the visual thinking because of that inward focus. And this is what the default mode network is kind of suggesting. The precuneus here, that hyperactivity for the autistic phenotype, is like the theater. It's the big screen. This is where everything is kind of being played out. And if you know about the default mode network, the hippocampus is heavily involved in associative learning and this high intensity of memory. All of this is involved here. And we'll get into that more in the second episode of the visual thinking. However, the default mode network manages the self-referential thought. But this reduced connectivity, such as the cingulum, and the hyperconnectivity of the visual parietal tracts, superior parietal and the occipital tracts, this boosts those imageries. Remember, just piece everything together that we've covered with the many columns and that heightened activity with the point A, point B being really fast. In contrast, the reduced STS connectivity, such as the arcuate fasciculus and the inferior longitudinal fasciculus, kind of limits the multisensory integration. However, this isn't necessarily the case the entire time or for every autistic, because this visual thinking, this live, realistic internal world is often mixing those sensories together at times. It's very fascinating on how it's just like a light switch. Remember the supplementing relationships. I can have full-on conversations about anything in this imaginative world. In the autistic brain, Grandin additionally writes, "My visual thinking is a strength, but social situations are harder." This highlighted default mode network kind of acts as a hub or a station that we can have all of this machine learning happening, and we'll talk about that in the next episode. We can build catalogs for categories in this intense learning, this accelerated learning that the autistic phenotypes have from way back to the Canner and Asperger kids to current. This is why people need to understand this visual thinking and this learning process so that the autistic phenotype, the autistic person, can do something that they love and can do and want to do. Why is that so difficult? I have something exciting. I want to introduce a product unlike any other product available. A highlight is the product from Daylight Computer Company created their product based on these factors. The Daylight computer is completely blue light free. It has no flicker. Short wavelength flicker is extremely harmful for our eyes and downstream biology. Light flicker is constantly turning our central nervous system on and off. Essentially, it is like going to a light switch and repeatedly turning it on and off. The problem is, blue light and LED light does this, and it is so rapid you cannot even perceive this in real time. The Daylight computer is the lowest stimulation and foremost for sensory sensitive users. It is no question that the alternative product, especially when used at night, do not address or consider this in their product. It is so toxic to human biology. Big tech corporations have patents on how their short wavelength implicate the human nervous system. And a bonus, despite daylight computer not having backlight, it is very functional for outdoor use. And of course, increased sunlight is always preferred. I am happy to offer a discount for the daylight computer. You can use the code AUTISM for a $50 off discount. Again, use the code AUTISM and the discount code for $50 off. See the link in the show notes to Daylight Computer Company, or just give it a quick search in your internet browser. Use the code AUTISM for $50 off. I would like to mention Chroma, lights designed for humans. Chroma, a Seattle-based innovator founded by ex-NASA and Air Force engineer Michael Shapiro, is on a mission to enhance physical and mental health with purpose-built devices, unlocking peak human health, cognitive function, and performance. Shapiro launched Chroma to restore the natural light lost to screens in indoor living, and delivers faster recovery, sharper minds, and better sleep. Their products hit hard. The Iron Forge speeds muscle repair with red and near-infrared light, while the Skylight mimics sunlight to boost sleep and energy. A standout, the Forge lamp, is a portable gem that fires 660 nanometer and 850 nanometer light to energize mitochondria and heal tissues on the go. Remember the 4 red light chromophores on cytochrome C oxidase, frequently talked about in the podcast. This is why. Their products are built with military-grade durability that have lasting impact. Chroma's tech fuses precision and power. Gallium nitride power supplies, smaller, cooler, and stronger than silicone, provide flicker-free light that's easy on the eyes. High-powered LEDs target key wavelengths for skin, tissue, and cellular health, with smart heat management for the lasting impact. Every design decision Chroma makes serves a purpose: to create devices that are precise, durable, and effective for improving human life. Remember, humans use different wavelengths of light for different functions of life. Remember when I ask, what do you think light is? Chroma designs with our biology in mind, from sleep aids and wound healing to mitochondrial energy, full body lights and blue light blocking glasses. They are US made and Chroma ships globally and accepts FSA and HSA payments. Use AUTISM at checkout for a 10% off discount. That's AUTISM at checkout for a 10% off discount. If you're listening to the podcast or listening to the episode, please feel free to leave a review or rating. In podcasting, reviews, ratings, and downloads are huge, and I very much appreciate your feedback. You can contact me on X at RPS47586, and we can have any discussion about autism. I'm always interested in your conversations about autism. You can email me, info.fromthespectrum@gmail.com. You can check out the YouTube page for all full-length videos and shorts and clips. And thank you for listening to From the Spectrum podcast.